Okay, so here we go, you guys, with the first part of chapter 3. I'm hoping there's only 2 parts, like I said, that 2 lectures that you'll have to sit through, and when you're looking for them on YouTube, you know if you've gotten both of them down. But anyway, so this whole theme is exchange, right? Transport, getting things from the environment, or even releasing wastes. And so a little bit of cell structure to start. And so cytoplasm is just the inside of the cell, and there's lots of different chemicals in there. And then your surrounding structure is your cell membrane. And many also have something else. Does anybody know what I'm talking about? What else surrounds cells sometimes besides the membrane? The cell wall, but not all organisms have them. We do not. Animal cells do not. Pretty much things that the cells that need to be more flexible typically don't have them. Bacteria does. The bacteria has a cell wall, and then plants, you guys think of, and fungus, and some protista, but not animals. And that's a bacteria right there. Oops, too fast. Okay, so the cells need lots of stuff, right? So just a little list here, of course, water, different ions, carbon dioxide for your autotrophs, and of course we have to get rid of carbon dioxide for everything that does respiration. Sometimes we only think of the plants as doing photosynthesis and us and animals doing respiration, but plants have to do photosynthesis and respiration. They make their sugars, and then they break down their sugars like we break down our sugars from our food. And then just overall nutrients, supply energy, building materials, some specialized molecules like hormones, and then we have our waste products such as ammonia, Okay, we're going to get to our membrane here. And the membrane is built of phospholipids. I have a goofy one still drawn on the board over here. And we mentioned them back in chapter 1, I think, right, with the chemistry, organic molecules. Some molecules can go through freely through the membrane, and some can't. Think about it. If these are all facing the insides of the membrane and they're hydrophobic, then is water going to go right through the phospholipids? No. You will hear, though, that water does pass freely, and I'll explain how it does. But it's not by coming in contact with the tails of those phospholipids. Another reason things can't get through the cell membrane might be their polarity. Again, remember, non-polars like each other and polars like each other. So water's polar, like I said. Size, just too big. Charges also. And the membrane, though, is called selectively permeable. Some things can go through and some things can't. So let's check this out.
Not all molecules are equally soluble in the membrane. The nonpolar phospholipid tails of the lipid bilayer tend to repel charged particles such as ions, but allow fat-soluble molecules to pass. Usually, the polarity, size, and electric charge of molecules determine whether they can pass through a membrane. The small molecules of nonpolar gases such as nitrogen and oxygen can pass through freely. Small polar molecules such as water, glycerol, and ethanol can also pass freely across the membrane. Larger polar molecules and ions would be repelled by the membrane. By limiting entry, a membrane is selectively permeable, which means that membranes regulate the exchange of materials in a very specific way.
So, when you talk about the function of the cell membrane, that's it right there. Plus, it's the barrier, right? I mean, which is still related there, but a lot of people want to say, it surrounds the cell, which is true. But that's related, right? Keeping the inside separate from the outside. you can't really say it does much support because it's not real strong. And you can see in a minute here, it's fluid. So it's very fragile. Now, transport proteins. And when you guys do your coloring, hopefully this really sinks in. So the membrane is not just phospholipids. There's other things in there. There's lots of different proteins embedded in between the phospholipids. And then some of them are transport proteins, and they allow some things to go through, like water actually goes through a transport protein. It's called a water channel. We already talked about that. Okay, so like I said, there's lots of different proteins. Some are known as glycoproteins. What does that glyco remind you of? Glucose. And so you'll see little carbohydrate chains sticking off the proteins. And then there's also glycolipids, and sugars can be attached also to the heads of lipids that are embedded in the membrane. And so what's their deal? Well, their function, they kind of have, since they have these things sticking out, it's kind of antennae. They receive chemical messages.
Membranes are composed of a lipid bilayer and proteins. The bilayer of phospholipids provides fluidity. Some proteins that are embedded in membranes have sugars attached to them, and they're called glycoproteins.
Really? It said actually, I'm going to start it over. Did you guys hear that? What gives fluidity?
Membranes are composed of a lipid bilayer and proteins. The bilayer of phospholipids provides fluidity. Some proteins that are embedded in membranes have sugars attached to them, and they're called glycoproteins. Sugars also can be attached to the heads of membrane lipids, creating glycolipids. Glycoproteins and glycolipids act as antennae that receive chemical messages from other cells. Proteins extending through the bilayer or embedded in the membrane's inner or outer surface make the surfaces asymmetric. Carbohydrate chains may be attached to both lipids and proteins. Membranes in animal cells also contain cholesterol.
Okay, and cholesterol helps with fluidity a lot. I'll show you a model here in a minute. But he said asymmetrical. What does asymmetrical mean? It's not symmetrical, right? Is that what I heard you guys say? So, and you can see it right here. We can't really see what's going on underneath, but it's different from one side to the other. But however, also very similar, as you can see, because it's built up of what? What's the main component? What's the main component? Phospholipids. So in my little box here, these marbles are representing just one side, and they would be the phospholipid heads, right? And you can see, got me popping them up, that it's fluid. The phospholipids are not attached to each other. So this is a nice model for what your membrane one side is like. And I can fairly easily get these phospholipids to change location. It's rarer, but it can even happen where they even swap sides. But that's going to be a quick thing. There must have been some push or something, energy provided to get them to move like that. But the point, moving like this is very easy, and that's why they call it a fluid. It's a fluid membrane. There's a word called fluid mosaic model used for the membranes, and that's because not only is there membranes in there, but there's other stuff jammed in there, embedded in between the phospholipids. I've got a little turtle here. And these things are the other components. What are the other components in the membrane? Read them. Okay, so there are all these other components that you can see there. Now, the other thing I want to point out is what the cholesterol and any other cholesterol does is cholesterol's a lipid, so it's a little bouncy, right, like lipids are, and that's going to help keep these even more fluid. Because if there weren't all these other things jammed in there, especially the cholesterol, The phospholipids would be more tightly associated, and then it's going to be harder for them to move around. Does that make some sense? So sometimes that cholesterol seems to be a real bad word, but you need cholesterol. Okay, and your body will make cholesterol from the food you eat, get some of it from the food you eat, but it's in that typical, has to be, that homeostasis. We can't have too much. We can't have too little. And there is some controlling there on our diets, especially if we have some genes that are genetics that aren't helping us very much. Some people are more prone to make more cholesterol from fats than others. So that is a real indicator to help. Well, it's a way of influencing, whether you get heart disease or not. But some people are just lucky, and their body doesn't make much cholesterol, just the perfect amount from the fat they eat. Okay, now we're getting on to how do things move. The membrane makes some sense, but what's going to make it move through it? Diffusion, you guys. Now, diffusion, you see it occurring like in this cup right here, and diffusion is just molecules move from an area of high concentration to an area of low concentration. I mean, you can just even think of a group of people, we're trying to get out of the gym, and we use energy, to get out, but we're trying to spread out. So a high concentration of people to a low concentration of people, okay? So, and you can see it happens with time. We're going to do a couple experiments later with this. Okay, now, for diffusion to occur, you need a concentration gradient. You need, like I said, a bunch of people compared to a little amount of people. The same thing with that cup. A lot of dye in one area and not so much in the other area. That's a concentration gradient. Don't let that word mess you up. Think about it. A grade, a hill, a higher spot and a lower spot. Concentration gradient. Now we're talking about the concentration. How much of something is there? Okay, so that's all it is. The concentration gradient is what drives the movement. And we're using free energy to make it happen. We're not having to put more energy into the system. Osmosis, hear me clearly on this, is simply a type of diffusion. It's specifically talking about what? Water. So water moves from an area where there's more water to an area where there is less water. Now this can get a little confusing, and it has all to do with how much stuff is dissolved in the water. That's the concentration gradient. If my big brother was making some Kool-Aid, and it was so sweet, he dumped so much of that powder stuff in there, there's not as much water as my dilute Kool-Aid. You guys following me on that? So it's not like where it makes sense the hill, where here's downhill, here's the top of the hill. What it is how much solutes are in the water. What's the solution like? Is there a concentration gradient that way? Because the water still wants to spread out. It still will move. Okay, back to that free energy. The free energy I'm talking about is just the movement of molecules. They're spinning, they're rotating, they're bumping into each other. Gas does this more than liquid, right? So things spread out through air quicker. And it's just because of the free energy. Okay? Jacob, you alive over there? Wake up. Okay, so now with osmosis, that's what's going to move the water into the cells or out of the cells. Now, cell wall. Cells that have a cell wall, they depend on osmosis. Well, all the cells do. But they depend on it for their turgor pressure, it's called. They want water to continually come in and kind of puff up that cell membrane, and it's going to smush it right up against the cell wall, giving it more support. Like if you think of a water balloon, there's some sort of support. Even though it's still fluid, right? But if you filled that up with water, then you can't bend your water balloon very easily. If it's limp and loose, you can bend your water balloon. Are you guys following me on this? So that's water pressure that is made through osmosis for organisms that have cell walls or single-celled things that have cell walls. Now, if you have something without a cell wall and water keeps coming into it, is there going to be a problem? What's going to happen to the cell? So let's check this out here. So here is your red blood cell, your animal cell. And if water is continuing to come into the cell, it's first going to swell, and then if it does not get equaled out, if the water doesn't stop coming into it through osmosis, what's going to happen to your poor cell? It's going to lyse or burst. That membrane's not that tough, right? Versus, look what happens with a cell here from a plant that has a cell wall. So it's giving it more support. And you guys see this with your plants at home. When you don't water your plants, they don't die instantly. But if they need water after a few days or maybe a week, what happens to the stems? Don't the stems fall over on your plants? Don't the leaves it's the stem part, right? Even the leaves actually kind of can shrivel up a little bit. That's because they need more water. Okay, now notice this concentration. You guys in the back, listen. Okay, here we have a high water concentration. To have a high water concentration, we must not have very much solutes dissolved in it. So that's actually called a hypotonic solution. We'll get to that in a minute. Here's osmotic or an isotonic solution where it's balanced. Same amount of solutes on either side, therefore the same amount of water either side. And then this last one is low water concentration. That must mean there's a lot of solutes dissolved in here, like my Kool-Aid. That was too thick with solutes. So the water is moving from the cell out. And you could see on where there's a cell wall, the cell membrane is coming off the wall. Here, no cell wall, so the poor cell is just shriveling up. Okay, so far I've only talked about passive transport. So you need to remember that. The two types of passive transport are, what did I just talk about? Diffusion. And then there's a secondary type of diffusion called osmosis. Good. And this simply means no energy needs to be used to make this movement occur. It's taking advantage of free energy through the concentration gradient. Active energy, obviously, listen to the word active, it must need what? It needs energy. So we're going to use some ATPs to do this. Okay, now I need to talk about, back to passive, I need to talk about another type of diffusion that's called facilitated. And that simply means that you're going to use one of those protein channels to get it through the membrane. So what the deal, a little bit more with that. So there's not just two types of passive transport I'm talking about then. There's a third one called facilitated. Now I have this one memorized thinking of a facility. Most facilities, I don't know about gels, but most facilities try and help people, like hospitals and schools and libraries, right? So all it is this has a helper protein that basically is like an open door for a particular type of molecule to go in and out freely, like water has one. Okay, now to active. Again, like I said, it needs energy, so we're going to use ATPs. So we're going to use ATPs. Do you guys remember the ATP cycle? We use an ATP, what does it become? ADP. It loses a phosphate, breaks a bond, gives us some energy, just a nice little amount. And then when the cell in a different part will be regenerating that, adding a phosphate, right? Okay, let's listen to this.
In simple diffusion, a type of passive transport, molecules pass directly through a membrane down their concentration gradient without any input of energy. In facilitated diffusion, also a type of passive transport, membrane proteins transport molecules down their concentration gradient without energy input. Active transport requires energy, such as from the hydrolysis of ATP, as well as transport proteins. Active transport can move molecules against or with the concentration gradient.
Okay, so this is important. This will definitely be a test question here. So these ones right here, passive, you can see is moving with the concentration gradient, higher to lower. This one over here is going against the concentration gradient because it's pumping these molecules in. Again, we have these transport proteins. They've done a pretty good job drawing them because they actually work like an enzyme kind of. The molecule fits perfectly in one side, and when it makes that connection, it changes the shape and lets it go out the other side, or the inside. Okay? So we could go against a concentration gradient, uses energy, this case uses a transport protein that requires energy like a pump. Do you see all the questions I could ask you, you guys? That was just on the active. I could do the same with the passive. Okay, hang in there. We're almost done. Now, for really large molecules, the cell membrane gets involved. And we saw this, didn't we, on that short little video last time with some funny names there. The pinocytosis, that's actually a type of endocytosis coming into the cell. That was water drinking, right? Then there was the phagocytosis of taking in big chunks of stuff, maybe food. You guys remember this from the little video? So again, now the cell membrane is engulfing or surrounding. This picture's 2D. It's a 3D thing, right? It's going to make a little vesicle if it's coming inside. You could see right here. The membrane actually makes, is a vesicle. It's put together by surrounding something, then migrating it, pushing it out. Maybe it's going along the cytoskeleton and out to the real membrane, where it actually then becomes part of the real membrane. Okay, so this will stop right there, and then we'll talk about our fish and our other stuff later. Okay.